All right. In the interest of promptness, we thank you all for joining us this evening. My name is Michael Myers. I'm the Chief Communications Officer for the Diabetes Research Institute Foundation. I just wanted to go over a couple of housekeeping matters, and then I will turn it over to Michael Burton. So this will be the first half of this evening will be a conversation, an opportunity to listen to two of our leading scientists, our scientific director, and our newest scientist, have a conversation about today's status of type 1 diabetes research, where things are headed, what the future looks like, and all the exciting news that's happened in recent months. I would ask you if you have any questions. We've had a number of questions submitted in advance that will be asked during the Q&A portion, the second portion of this evening. But if you have additional questions, please feel free to submit them via our chat room here, and we will answer as many as we can during the course of the evening. If we don't get to your question, we will have a copy of this session is being recorded. We will be sending a copy of the recording along with answers to any questions that were unanswered during this evening's session to each of you, to everybody who's registered for this evening. And I thank you. So with that, I would like to turn it over to my friend, colleague, and our Chief Executive Officer, Michael Burton.
Good evening, everybody. Thank you so much for joining us. As Michael said, I'm Michael Burton, the Chief Executive Officer of the Diabetes Research Institute Foundation. It's a pleasure to be here with you tonight, and we're very grateful that you've joined us for what we believe will be a very special evening. We are joined this evening by our scientific director, Matthias von Herrath, and our newly recruited scientific leader, Dirk Homann, who will be the focus of our attention this evening, giving us an opportunity to really introduce him to you. So we're excited about the program ahead. These are, I think, extraordinary times of progress and promise in the field of diabetes research and care. I don't think there's any question that historically the Institute has had a tremendous impact on the trajectory of how we treat this disease, how we understand it, and how we translate those understandings into new therapies. The Institute has a proud history, no question about that. And we have our fingerprints, so to speak, on some of the most important discoveries in this field. And that's a proud past, but I have to say that we are focused like a laser on our future. And Dr. Homann's recruitment and his joining this community of ours, I think, really speaks to just how exciting that future is. So I'm going to briefly introduce Matthias and Dirk, and then hand off the program to them. They'll have a discussion, and then we'll move into Q&A. So we were very fortunate 2 years ago to recruit Matthias von Herrath to the Institute as our new scientific director. Matthias is a world-renowned virologist and immunologist who has made a significant impact on our understanding of diabetes and how we treat it. So it was, I think, a real coup for the Institute to recruit him from La Jolla. And in the 2 years that Matthias has been with us, I think he's really brought a tremendous amount of focus, energy, certainly talent, and vision to the Institute. So we could not be more pleased with his joining us 2 years ago and all the impact that he's had since. One of the most important things he's done, I think, is recruit Dirk Homann, who, again, is joining us this evening. Dr. Homann is a physician scientist with a deep passion for understanding how the immune system works, especially in autoimmune and infectious disease. Before joining the DRI, he worked at Mount Sinai in New York City. His training and academic journey have taken him through world-renowned institutions in Berlin, Boston, Paris, and La Jolla. Dr. Homann studies a key part of the immune system, T cells. These are the cells that normally help protect us from infections, but in T1D, they mistakenly attack the insulin-producing cells in the pancreas. Dr. Homann's research focuses on how T cells develop, how they remember what they attack, and how we might guide them to protect the body without causing harm. His work is helping us uncover new ways to prevent and treat autoimmune diseases like T1D by better understanding the immune responses at its core. We are thrilled to welcome him to the DRI family and to welcoming him here tonight. So with that, gentlemen, I'll hand the program over to you.
Thank you very much, Michael, and I hope everybody can hear me. I'm super excited that Dirk is joining us, and Dirk and I have had a long journey together, mostly scientific. Started around, I think, 1995, if I'm correct, throughout our whole careers, and we have had ample of dialogues and discussions about science that we have probably both enjoyed a lot. We also play music together. I should say Dirk, as opposed to me, is a professional musician with a whole professional musical training. So on that team, usually Dirk plays the piano much better than I play the violin, but nevertheless, we have a lot of fun. And staying with the same team, Dirk's research now, that's why we are so excited having him join the DRI, probably has in many ways surpassed what we have done in the early days. A big topic will be today, and I want you to ask yourself this: when you look at a disease like type 1 diabetes in humans, a very principal question is, how do you even understand how such a disease develops? And without the understanding, it's difficult to be fast enough and do the right therapies. And about maybe 15 years ago, the whole field really experienced a seismic shift and moved from studying mouse models, which can have advantages, but disadvantages, mice are not humans, to studying human pathology. So this will be a big theme, and it came through the National Pancreatic Organ Donor Consortium. And Dirk is a leading scientist in pathology and biomarkers, and we will discuss this today in much more detail. But my first question to you, Dirk, is why are you excited to join our DRI family? What are your expectations? And then we will segue into the actual meat of your research and explain a bit what is so exciting about it and why we think it's super important.
Well, thank you, Michael and Matthias. I'm excited to be here. Hello, everyone. Thank you for making the time and joining us tonight. I received training as a physician, and then the vagaries of career development. I decided to pursue research. I did this together with Matthias and our common mentor, Michael Oldstone, in California at the Scripps Research Institute. And I was set on a path that I was highly intrigued by. However, over the years, my past is in many ways catching up, and the original motivation to do something that is for the benefit, for the well-being of patients, is prompting me to reconfigure my research a little bit. So while I do not practice medicine anymore, I seek to be more involved with aspects of research that have a more tangible, more direct benefits for patients. And that's where the DRI comes in. The infrastructure of the Institute, the cross-disciplinary approach, and the opportunities are almost unparalleled. And I think at this later career stage for me, it is it's an exciting place to actually put some things more into practice after decades of basic research, which has its role, it is important, but I want to be part of a more applied aspect. And I think the environment at the DRI will give me that opportunity. And I have over the years gotten to know many of the investigators at the DRI, so I know what I'm getting into, and I very much look forward to it.
Well, we are super excited, Dirk, and I can promise you, from my perspective, we can give you honest, open dialogue and many discussions about science, sort of in the DRI family style. And I think that's really important nowadays. And it's going to be super exciting, in addition to some music together, which I'm looking forward to. Now, coming to your actual work, and particularly the understanding how the human disease, type 1 diabetes, evolves through studying pancreata, donor pancreata. I know you have taken this much to the next level. We used to do this in a more stationary way, as it is by nature, that you have a tissue picture, you have to imagine, and you can look for certain cells. But that gives you kind of a snapshot of certain cells, and maybe not at the detail you need. And definitely it doesn't tell like a longitudinal story, but we want to have longitudinal stories at the end about what happens first and what happens second and leads A to B to C. So tell us a little bit about what excites you, where you are with the studying of histopathology, and how you see this going in the future. It's a big question, so please take your time.
But I jump on the prompt story because this is the business we are in. We tell stories, and we tell these stories is based on experiments we perform in the lab, observations we make, and we string those information, those observations together in a fashion that hopefully results in a cohesive and compelling storyline. As chance will have it, we have just right now 3 papers that are the fruit of a multi-year effort, each either in press or submitted, that reflect the spectrum of my interests. And I'll start off with what you asked about with pathology by going back more than half a century, a story that involves the legendary physicist, Nobel laureate, and merry prankster Richard Feynman. Some of you may have heard the name, even read one of his books called Surely You're Joking, Mr. Feynman. It is very much worth your time should you be curious. Feynman was asked to give a keynote address to an audience of biomedical researchers. And one of the questions that he got was something along the lines, Professor Feynman, what is next? What should we be doing? And he quipped allegedly that is, well, just look at the damn thing. And this is exactly what we've been doing, and in many ways what we should have been doing all along. And I will just mention another little bit of history here. The notion that type 1 diabetes is an autoimmune disease was put forward in the 1960s. Essentially by the work of Willy Gebz, a Belgian physician who took tissue sections from the pancreas of individuals that had tragically died following onset of type 1 diabetes and observed these tissue sections under the microscope. The idea that this is an autoimmune process that initiates the disease and promotes its progression was then consolidated in the 1970s with the discovery of autoantibodies and linkage with genes, in particular the so-called HLA class 2 genes. At the same time, by pure coincidence, a mouse was discovered in Japan that since has been named the non-obese diabetic mouse that developed symptoms not unlike human diabetes. And by 1980, this mouse was a fully established disease model. And ever since, I've crunched the numbers recently, or ever since, there has been at least one article on the NOD mouse every single day, including weekends and holidays, which means that there's a lot of stuff we know about the non-obese diabetic mouse, and none of us as individuals actually knows the entire literature. So what this prompted is that researchers diverted their attention to a small animal model because it was easier to study at the expense of human research. As it turns out, and I'm sure you all are aware of that, The pancreas is typically not biopsied, so we cannot take a look under the microscope of a tissue section. And it is difficult to procure because of all the digestive enzymes. It has a tendency to self-digest. So this takes an enormous amount of effort that many investigators simply shied away from. And it was individuals such as Alan Fowlis in the UK who more or less single-handedly with his team collected pancreas from deceased individuals, from donors. And by looking again under the microscope, he reported some of the fundamentals that still inform and inspire us, that make type 1 diabetes a very distinctive disease at the tissue level. So with that long introduction, coming back, the type 1 research experienced, especially histopathology, experienced a transformation when around 15, 20 years ago, a consortium was founded here in the US, the so-called Network of Pancreatic Organ Donors with Diabetes, that procured, processed, and distributed pancreatic tissue specimen to interested and affiliated investigators. And what that did was finally what science is so good at. It democratized research. Many investigators who never would have been able to get their hands on these precious tissue specimens now could do their thing. And that has revolutionized the way we can do science. In the medical sciences, oncology is typically at the forefront of technology development. That's where the money is, that's where a lot of talent is. And because type 1 diabetes is less prominent than cancer, the application of these technologies usually lags a little bit behind. So we actually have the advantage of choosing technology platforms that were established as fairly robust and apply them. And that's essentially what we did. We used a state-of-the-art tissue staining technology that allows us to visualize a multiplicity of proteins in pancreatic tissue sections. And one of the key challenges in evaluating pancreatic tissue sections is that much of the tissue is not what we primarily interested in. It's exocrine tissue. 98% of the pancreas or so is exocrine. We are interested in the islets. That's where the beta cells are. And that's where we are looking, trying to understand how these beta cells are impaired and destroyed. And because these tissue sections are relatively large, perhaps the size of a thumbnail or even bigger, it is difficult to manually evaluate that. So we used machine learning or artificial intelligence, if you will, to develop image analysis strategies that allow us to capture a great amount of data. And to make that long story short, and to finally come to the point, what we learned by applying these automated analysis strategies is that we can capture everything that we already know. This is good because we want to have some reassurance that what we are looking at is meaningful and robust. Based on that, we believe that we have discovered a whole slew of new things in the not only diabetic pancreas, but importantly in the pancreas of donors who had 2 autoantibodies. We know that these individuals, had they lived longer, would have developed diabetes, even though we do not know if it would have been 6 months or 20 years. That we cannot tell yet. But at the population level, we know that these individuals would have gotten diabetes. And what we have found is essentially that by using these more sophisticated image analysis strategies, we find that many of the changes that are so typical of type 1 diabetes at onset in the pancreas, they are also present in these autoantibody positive individuals. What that means is that in many ways, type 1 diabetes is a disease that develops like you would expect it to develop. That there is a prolonged period that is not clinically apparent. Individuals do not know that they have it, but it is already inscribed in the pancreas. So in many ways, I'm excited about this because it means that at least looking at the histopathology, that type 1 diabetes behaves like a lot of other diseases, in that it slowly builds up, and once it crosses a critical threshold, then it becomes clinically apparent, and people will have to go to the ER, and they will have to, their lives will change with that diagnosis forevermore.
Dirk, what excites me particularly about this line of research is that AI is now getting involved. And in a way, it's telling a story from having stationary pictures through various stages, we presume, at the disease, into then a fully timeline cohesive story. I'm very interested in understanding from you what happens, or what is your opinion? What is your AI's opinion? What happens early in the disease? People always ask questions about the chicken and egg situation. Is it the beginning what sets diabetes off? Is it a dysfunction of the beta cell, or is it the dysregulation of the immune system? Who comes first? Maybe you can speak to that a little bit.
Of course, I don't have an answer, so I have to say yes
to
both options.
But maybe the AI has already suggested things.
So what we can tell is that many of you will have heard the term insulitis. And that is an expression that captures an inflammation of individual islets. And that's a hallmark, a pathological hallmark in the pancreas of individuals with type 1 diabetes. What we found is that while this is certainly helpful, other islets that do not have the insulitis features are also affected. So meaning that the disease process is remarkably tissue-wide. It is not restricted to those areas that have these insulitis features, but it is all across the organ. And that actually makes a lot of sense. But we hadn't looked at the other non-insulitis areas because there was nothing obviously wrong. Again, it required the more sophisticated analyses of every single islet and every single tissue sections to come to this conclusion. We also find that in autoantibody positive individuals, there is an influx of T cells, meaning that the number of immune cells that are present is higher than in individuals that are not pre-diabetic, that we consider non-diabetic at the time. Again, remember that all the tissues we can look at are donated by individuals who tragically died and who graciously donated their pancreata for research purposes. So we take this quite serious, and we want to make the most out of these precious tissues at our disposal. But the recruitment of immune cells in the pancreas in the preclinical stage is actually quite interesting. And again, to make a complicated story somewhat short, we believe that initially, so you may have heard that what is quite curious, what has become apparent over the past decade, is that the sheer number of T cells that recognize beta cells that you find in the blood of individuals with type 1 diabetes and those that don't have diabetes and never will get diabetes. The numbers in the blood are pretty similar. That's astonishing because when this was first looked at, we all believed that individuals with type 1 diabetes must have higher numbers. So what we believe now, what happens is that in the preclinical stage, all immune cells, especially T cells, are recruited to the pancreas. And there, simply by having more cells, even though the frequencies are the same as in healthy individuals, this is setting the stage. And at later stages, when individuals actually get type 1 diabetes, and we can look at the pancreata, we find that now they are enriched for those T cells that recognize and presumably destroy the beta cells. Again, this comes down to a very simple development. We cannot yet say what the initiating events are, but what we do know is that beta cells play a role in that beta cell stress can lead them to become more vulnerable, and that T cells that are in principle capable of destroying beta cells that are found in healthy individuals, as well as those that are pre-diabetic and diabetic, that they can be recruited, and when the two come together, the specific T cells as well as the vulnerable beta cells, perhaps stressed out for a number of different reasons, then the disease can be initiated and can progress towards a clinically relevant stage.
That Dirk, this is really important, I think, and let me take the liberty to put this in some context for the audience we have here. So we talk here about the history and how diabetes develops in patients, and it is probably the realization that you need the pancreas, the beta cell, as well as the immune system to misbehave in context to get this disease. That's therapeutically very important, and it's very important for prevention because you end up in the concept that you need to have probably combination therapies, not immune, but also to strengthen the beta cell health. Now, you will ask, how is this relevant if you have an islet transplantation? Islet transplantation is highly relevant because stem cell-derived islets, when they come in the body again, they will be, in the worst case, subjected to the same scenarios where the immune system becomes again curious about them and starts attacking them. Understanding this is of fundamental importance, I believe, and that's why I believe your research is so important. We always say there's no cure without the prevention. There's no prevention without a cure, because these two processes are in a way linked at the hip. I have one last question for you, Dirk. Maybe a little bit pointed question. I often ask, maybe not too popular, but maybe important. Before we go into the general discussion part of our chat here, you were talking about that the number of cells that can see and attack islets has been similar between individuals that are healthy or have disease, and there's an attraction and then maybe an augmentation phenomenon in the pancreas. We have done in the past several longitudinal studies where we try to understand whether we can find markers so a patient would know on what precisely point of the trajectory they are to develop diabetes. And you work on biomarkers. In our longitudinal studies, it has been very difficult to find a marker that would tell you, okay, they would say, Matthias, you have these markers, you will develop now diabetes in a year, which would be an answer that we are all looking for, right? Where are you in your thinking about your biomarker studies that are very well known, very sophisticated? How is your hopes in that? And just very brief you can be, and then we go in the next part of the hour.
Despite a lot of disappointment, many people, many smarter than I, have thought about this for quite some time, and the results have been disappointing. So we launched a major effort, and actually much of my external funding comes for these particular efforts to find biomarkers in the peripheral blood. Much of the potential success comes from the cohort of blood donors. So if that is well selected, then the probability that we find something relevant is increased. So we've been very fortunate to work with the TrialNet Consortium. Some of you may have heard of them. They organize interventional trials of various kinds. But the crown jewel in their collection is the so-called natural history study, where they collected periodically blood samples from individuals that are, at high risk and from control donors. So there is a large collection of blood and serum samples that can be analyzed. And because the resources are limited, the TrialNet wanted to be sure that studies that make use of these samples make the best possible use of this. We've been lucky to get access to both blood cells as well as serum. For the blood cells, we collaborated with a group in the UK, with Owen McKinney. We looked using a technology called mass cytometry at the protein expression profiles, and Owen McKinney in the UK looked at the transcriptome of these cells. And this is one of the papers that is currently in review. And what we find is that, again, at a very early stage, prior to clinical onset, we do see, in fact, robust, what we call signatures. There are changes in immune cell composition and in immune cell characteristics. These are subtle changes. They are not striking. If they were, others would have discovered them a long time before us. But by combining a remarkably well-designed study cohort that's all been done by TrialNet with state-of-the-art analysis technology, we now have something like a signature that is present years before the individuals develop diabetes. It's complicated. It's not a single marker. So we need to do better. And that's where the second part of the study comes in. From the very same blood donors, we have the serum. And we put together, my co-principal investigator, Kate Speak at the Benaroya, and myself, something that is arguably the largest serum study ever conceived in the type 1 diabetes field. And we recruited half a dozen or more colleagues who are specialists for very unique analysis strategies. We use mass spectrometry, we use flow cytometry, we use phage immunodisplay, and a host of other technologies. And we cast a very wide net. And we hope that it is wide enough to tease out what may serve as a future biomarker or a combination thereof. So we covered the cells as well as the soluble factors in the serum. This study will be done within the next 3 years or so. But we will also, and I think this is important to mention, we will make data publicly available whenever we have it. Meaning that we do not presume that we will be the only ones who understand these data. We will actively solicit input from others and give them the opportunity to analyze all these data by themselves, because the effort is not to publish a great paper, but the effort is to find something, and we can only do this if we not only cast the net wide when it comes to the analytes that we quantify, but that we equally have a diversified spectrum of colleagues that take a look. And we are in this together in so many ways, and only by truly leveraging this opportunity can we hope to generate insights that may make a difference.
I agree with you. We need, we know we need large data quantities for prediction in the AI to work. We have seen this with ChatGPT and now with image prediction, and then also the ability to write a programming language. I mean, this field has made a huge jump because of the number of data, among other factors. And I'm with you. I think if we all collaborate together, maybe we can find these signatures. It would be so important for the field, not only in prevention, to know precisely where on a disease course a patient is in a given patient, but also after eyelid replacement to know whether there are early changes, so you can anticipate whether there's a heightened risk of losing the graft. Because the earlier you can do something, usually in the human body, the better we are off. If we come late to the party and try to treat something that's already established, it gets much harder, as we all know. I want to turn over to Michael at this point, because there's many questions in the chat, and both of us can take some time to answer the burning questions here. And I'm super excited that you're joining us, Dirk. We will have a productive and also fun time together. This I'm really looking forward to.
I officially started on Monday. I'm back in New York right now because I need to supervise shipment of equipment and reagents. But I'll be back in Miami on Tuesday, and then we'll get started.
Thank you, gentlemen. You guys are making great science and great music together, so we appreciate it. We've got a number of questions that have come in, and so try to be as brief as you can in your responses, but I want to make sure we devote the appropriate time to them. And as I said earlier, for our audience, any questions that we don't get to or any questions of a personal nature, I promise you that we will be responding in follow-up in the coming days with our scientists involved in those responses. So let me start. First question that came in, it looks like you study many individual cells from the pancreas to look for clues about how diabetes develops. What is the biggest difference you found when looking at all of those cells?
That goes for Dirk.
Yeah, this goes along the lines of what we talked about a little bit earlier. So there are many different things to look at. We decided to cast a particular focus. And one, as I mentioned earlier, one of the unexpected findings was that islets that do not have any immune cells that attack them, at least at the time the tissue section was obtained, they already show deficits when it comes to beta cells and expression of beta cell hormones. As it turns out, one beta cell hormone that is not insulin, it's another hormone. It's called amylin. Amylin production is shut down very early on. We did not know that before, so it may be something to take a deeper look at. Our particular study was very much focused on the endocrine cells in the islets. We did not look with granular detail at the immune cells. That is something we are in the process of doing. Others are doing that as well. So there is still much to be learned. But even though I am an immunologist and my passion, my first love is T cells, for this particular study, we decided to take a closer look at the endocrine cells in particular.
Another question here. What makes studying the pancreas so challenging? And how do the new technologies that are offered now help you overcome those challenges?
Well, the first big challenge, again, as alluded to, is procurement of tissue. These are all donor tissues from individuals that died before their time. And we need to honor the gift that we receive by being allowed to work with these tissues and analyze them. And one of the ways of maximizing outcomes is to look at everything, meaning that we should not just look at some islets, we should look at the entire tissue section, at the entire tissue specimen. There are other formats of tissue preparation that are also important for studies, such as so-called tissue slices, etc. At the same time, modern technology platforms give us the opportunity to not only look at one thing, but at many different things at the same time. So we collectively, as a community involved in this type of work, we strive towards maximizing what we can get out these very precious, these very limited tissue sources. But one interesting development just this year is that several groups who do this type of work have used many more donors that they could get their hands on. So we are right now, this year, in the midst of a process that eventually will allow us to better understand differences between males and females, between people who have maybe obesity and have not age of onset, duration of disease, ethnicity, etc. These are all factors that figure into the natural history of type 1 diabetes. But thus far, we had too few donors to tell compelling stories. And this is happening right now. So this is actually 2025 will be an interesting year because there will be at least half a dozen milestone publications that I'm aware of that will be coming out from various labs.
Thank you for that. You mentioned modern technologies. What about the use of even AI-like approaches? How is this changing how we understand disease and develop new treatments?
It's a long discussion, and I'll keep it short. Then Matthias can chime in there. The general public became aware of AI with ChatGPT, and this is undoubtedly, a remarkable advance. But AI has been used for decades, and for certain niche applications. So machine learning has been part of what Matthias and I have been doing for a long, long time. And with newer capabilities, these approaches will give us better possibilities to discover patterns, etc. And this is one of the directions that Matthias and I have been talking about, that we want to be the DRI at the forefront. We need to build the architecture for that, the infrastructure. We need to hire talent, etc., to allow us to do this on site. But in many ways, what we use for this particular project I talked about, this is in many ways classic, AI. This has nothing directly to do with ChatGPT or so, but it is related, and obviously we try to take advantage of the remarkably expanding possibilities that machine learning, artificial intelligence provide us with. Matthias.
Yeah. Can you hear me?
Yes.
Good. Yeah, I can add one thing to the theme of AI. AI needs large amounts of data, and it needs very good data. And I think at the DRI, in our thinking, the generation of very good data is probably something we all need to work together. And what becomes particularly paramount and where we would like to lead the way is that in science, in the scientific method, we need positive data of things that work, and we need negative data of things that didn't work. And we are very passionate about paving the way for scientific enterprise, a field, publication field where we publish positive and negative data, so that actually AI systems later on, including the data we will derive from the pathology, can have the whole picture, so to speak, because otherwise you can't blame the AI if half of the picture is missing. And some of you might know that in science publications we have a positivity bias, and I think to become more centric and publish positive and negative data and very solid data, gold standard vetted data will be of paramount importance to take our scientific enterprise into the next decade. And we are very passionate about that. And it links to these AI approaches in an intimate way. Michael, back to you.
Thank you. We know that some changes in immune cells can be detected before someone even shows symptoms of type 1 diabetes. Could these findings eventually help doctors know who is at highest risk and maybe even stop the disease before it starts?
That's the point. That's entirely the point. And the better we become in predicting that something is going wrong, ideally, like we discussed, to the blood, the better we will not only become to prevent the disease, which I'm extremely passionate about, but also early anticipate. If you take a stem cell-derived islet graft, even the newer grafts that might be immune evasive, I think there were some questions in the chat, and genetically modified, I agree with that. We need to anticipate early on whether such grafts become rejected. And it will be very important, because if you come late, so to speak, to the party, and you try to do something after half of your graft has been destroyed, it's already too late. We need to detect the earliest signs. That's sort of the finesse of biomarkers, and probably in the future in conjunction with AI. There was something that gave me a lot of hope. There's a company a friend of mine has who are working with biomarkers in oncology. And they now have a marker-driven system similar to what Dirk is talking about, to predict whether, if you have a tumor, you might have a recurrence, which is obviously of paramount importance. Any of us would want to know this if we are afflicted with cancer. And that seems to be quite successful, and they have now an approved and validated test for this. So I'm quite hopeful that in conjunction with AI and large, good amount of data, these type of barriers can be broken through, and it will accelerate the path to ultimately have curative approaches for type 1 diabetes, because early prediction will have to be essential for that.
And just to add to this, it has to happen at various levels, and it may well involve state-of-the-art artificial intelligence approaches. For instance, there's the interesting niche developments for unusual diseases. The term drug repurposing is used to describe the use of approved drugs for a purpose different from what they were intended for. And there are several groups throughout the world that, in dealing with diseases for which there is no ready cure, these are often very rare diseases, so there is not a lot of research, that they train AI systems based on the available literature that may, span decades and include papers that most of us would never have, bothered to read. And these AI systems come up with suggestions. And in several cases, unusual drug treatments led to amelioration of symptoms or even cures that were simply not conceivable by using traditional approaches. So this is another example how AI, in a very different fashion, can be very usefully and effectively integrated into developing approaches to better therapies.
Thank you for that. Another question. In the past, DRI was quite optimistic about encapsulation and artificial pancreas-based cures. Are they no longer viewed as viable cure-based approaches by DRI? Could the audience get a brief overview of your assessment of this and of possible cure-based approaches that are still being investigated?
Yeah, that is, of course, a very important question. I can take this. And as many of you know, especially Camillo Ricordi and Alicia Tomi and many others have worked very intensively in this area. And some of you might know that the field had a setback because just a few days ago, the Vertex trial reported with devices that they're abandoning this route to the clinic because of lack of profound efficacy. So here's where the field is, and it's actually in a pretty promising spot, in my opinion. It is indeed true that devices that have a barrier between the islets and the blood system they have not had a stellar road of success, any of them. That included the occlusive ViaCyte devices and many other devices. We think it's probably because the islets need so much blood supply. They literally drink straight from the water hose, I always say. And Joanna Almaca has very nice data with Parasite showing how they even turn off their own spigot, so to speak, having enough blood supply. I think we just can't give them enough of this in a membrane closed device where the islets sit on one side and the blood is on the other side. So where the field is that we need to combine, I always say, find a sweet spot, but by having a device that allows vessels to grow in, but holds the islets. And there are now many advances at the DRI, also collaborated with other investigators in this area. So you have a holding tank, the vessels still come in. Then you will say, well, but if the vessels come in, the immune system can reach the islets. Also, this is true. But we will have to combine this, and that's why I talk about the sweet spot. We will have to combine this with islets that go in such devices that can avoid immune recognition. And we have at the DRI now a major effort making mini organoids of islets that we do in our GMP facility, where we can test different modifications and see how much immune modification is needed to have the islets evade the immune system. This type of sweet spot, I think, is going to bring the whole field of islet replacement to the finishing line. So I would say this failure of occlusive devices is not entirely unexpected, although I thought the design of the device that Vertex has, at least from their published patents, I don't know any other details, looked very interesting. As disappointing as it might be that we cannot bring these barrier devices easily to the clinic, there are many other avenues that we are focused on that look quite promising. So that's where the field is. Having a holding tank where you put islets rather than putting them just in the liver is better because you have a better sight on the organ graft. You can monitor it better, and if something will accelerate the path to the clinic. Sorry, that's a big field. It was a bit of a longer answer, Michael, but that's a fundamentally important field.
A quick follow-up came into that. Do you think there are still possibilities for islet cell therapies that would not require any immunosuppression?
Absolutely. That's what we're aiming for. That's a huge, large reason for the existence of the DRI. We need to have cell immunotherapies where we don't have full immunosuppression. And for me, the sweet spot is you have a holding device, you put cells in there that are partially at least immune evasive, and as a consequence, you need a much milder form of immune modulations. And there's therapies that can be supportive of beta cells, for example, that could fall into this class based on DRI data, maybe even double graft survival. And that's where the sweet spot will be. I always give also the analogy of flying an airplane. It has to operate in an optimized space. In the case of the airplane, of speed and wingspan and these type of things. And it will be the same for the islets. They will evade the immune system. They will be in a holding tank, and you take some of the immune system back. And this sweet spot will come then paramount to a cure for patients over time, because it becomes extremely well tolerable. So it's a good question. That's what we are striving for. We need to achieve this to ultimately have a cure for the disease. That's where it's going.
Thank you for that. Another question. Can you share your thoughts about autologous stem cell treatments to produce islet cells that are then edited to be hypoimmune, as was done in a recently released study in China on a young woman? And is this something that will be pursued at the DRI?
Yep, that's what we need to pursue. We need to overcome two things, and this will be done in our GMP facility. One is we need to make sure that they're called iPSC-derived approaches, that the final product is safe and doesn't have untoward cells in it. And we need to make sure we only make the minimum necessary genetic modification to make these cells immune evasive enough. If they are autologous, we are getting a big head start, because what people call alloimmune rejection, where one immune system, let's say yours, would reject my cells, that's off the table if it's your own cells. So we get a better start. But that the system, and Dirk spoke about this in much detail, of the autoimmune T cells, they will still be there, and they will have memory, as Dirk alluded to, and these cells will still recognize such grafts, so they need to avoid this. Again, this is a sweet spot finding. We think that the organoid platforms we have established now with a Swiss company in Sphero in collaboration, that allow immune attack of islets in a very reproducible fashion in vitro, will help us to define this sweet spot field, as I call it. And that's where this is going to go.
Thank you. Is the DRI involved with gene editing to delete the signal that the beta cell would send to the T cells?
Same. That's the same answer. The main signal that the beta cells send to the T cells early on, Dirk was talking about the early changes, is upregulation of a molecule called MHC class 1. And it's a hallmark pathological feature of type 1 diabetes. That class 1, that is usually in healthy beta cells and islets very low, goes up for reasons we don't understand. We definitely don't want this to go up on transplanted stem cell derived islets. It's not going to be good, because the immune system is almost like a Harry Potter cloak is pulled off these islets. The immune system is going to be able to find them. So that's these obvious genetic modifications need to be done. The field knows this. Many are also working on this. I think these modifications are going to get us to the finishing line ultimately.
Thank you. Can you talk a little bit about the pros of JAK inhibitors in delaying the onset?
There's no doubt, and this was published in New England Journal, that there's the JAK inhibitors work. I would think, ideally, that we have a treatment, either you only use the JAK inhibitors for a short time and then have a maintenance therapy. That is one possibility. So you induce with a short course of a JAK inhibitor, and then you have a maintenance therapy. I think the equipoise for type 1 diabetes as well as islet transplantation is not to permanently stay on these, because of the advances in insulin therapy. At some point, hopefully GLP-1s, and also then associated the devices, pumps and closed loops. So we can only envision for immune modulation and suppression that's systemic to have a short course therapy for this disease. That's my opinion.
Dirk, I want to focus this to you. What research advances over the last 12 months excite you the most as you focus on cure and prevention?
Actually, what I said earlier, the papers that will be coming out this year on better understanding the early changes in the pancreas, in the disease process. There's much happening in the field of biomarker discovery, and we aim to play a role in that space. But if I may, I would like Matthias could speak with much more insight about the actual projects pursued at the DRI. So I'd like to tie this together with a perhaps more philosophical perspective, just briefly. Science operates in a unique fashion, and the exceptions are the Manhattan Project, Moonshot, for instance. Curing diabetes is arguably more challenging and more complex. And what science is good at is exploring a large variety of different ideas and approaches that we believe may make a difference, but ultimately do the work in the lab to see if our predictions actually hold. And often they don't. There's a famous word by Sam Beckett from one of his later plays. It goes something like, ever tried, ever failed, no matter. Try again, fail better. And that's what we are doing. we look at various aspects, and Matthias's work, my work, are a small part of that. We look at modulating the immune system. And the goal is to develop some sort of immune intervention that is as effective as possible, while at the same time being as mild as possible, because we do not want to impair the immune system and its ability to fight infectious diseases and cancer, etc. Right? We are investing into pharmaceutical approaches to spur regeneration and growth of replication, proliferation of beta cells. There are advances in device technology, the encapsulation. There are hurdles and challenges, of course. Stem cell derived endocrine cells, in particular beta cells, but other endocrine cells as well. You know, the generation of entire organoids in the dish. This is all possible. So likely no single direction will lead to a breakthrough, but rather a combination thereof. And when that will happen is difficult to say. The obvious example, is something we all lived through. It's the pandemic and the remarkable speed with which mRNA vaccines were developed. Just as a brief reminder, the virus, the actual genetic sequence of the virus was uploaded by Chinese investigators in January 2020. Days later, the vaccine design was in place, which led to a rapid development, scaling, and a year later, vaccines were available. Some people were suspicious. They said, something must be, wrong here. This is not possible. That does not take into account the fact that the key developments that made this miracle possible are based on decades of research. One was using mRNA for vaccines. That's been tried out almost 50 years ago. There was a breakthrough in 2005 that made all the difference. The two people who authored the paper now have the Nobel Prize. The other important development was lipid nanoparticles. This is the vehicle with which to actually carry the RNA to the cells that are important to prime, to generate an immune response. That was developed in Canada, also over decades. And the third important factor was research on a completely different virus, a respiratory syncytial virus that, however, shares aspect that allowed investigators to focus on a particular part of the coronavirus, namely the spike protein, and they knew immediately how to modify it to make it into an effective vaccine. So this is based on research that happened, outside the purview of the public, because this was niche knowledge. This was not something that was worth reporting on primetime TV. But because it happened, it was supported. We were lucky that in 2020 all that came together, and it could create a revolutionary breakthrough. I believe that something like this eventually will happen in type 1 diabetes, but we just don't know what aspects will make the differences. So despite a lot of failures and disappointment and setbacks, it is important to maintain a diversified approach, to look at many different things, and eventually the probability that they come together and will then bring about a breakthrough is there. It's just difficult to predict when that may happen and what actually may be part of it. But again, this is very much the nature of scientific investigation. Of course, we have ideas and predictions and hypotheses, but more often than not, they turn out to be different. And this is exactly why we do the work, why we ask, so to speak, in nature to provide us with answers so that we can do it all over again, but better.
Well, that's why at the DRI, we always say we need to celebrate all our small victories, because many small victories will amount over time to a fundamental change and maybe even accelerate the curve toward change. And then we will approach the territory of having a cure for a disease. That's how I believe this is going to come together. Michael, back to you.
Thank you. Thank you for that. A couple more questions. Then I just want to let our audience know we will keep the line open following the call for another 10 minutes if you want to submit any additional questions via chat, and we will respond to them as part of our follow-up in the coming days. And excuse my pronunciation on this, gentlemen. Islet cells are normally grouped together in the islets of Langerhans, and some studies show that transplanting islet cells in a concentrated group help to prolong their life. Is there any evidence of the importance of the Langerhans?
Well, so Langerhans was the one who discovered these islets hanging together. There's no doubt that when we make stem cell derived islets, we have to combine them together in the whole context of an islet. And even more so, we probably have to give them some cells that are sort of, even in the normal context, peacekeeper cells or surrogates of peacekeeper troops that we deploy together with these islets. That's why putting them in a holding tank is important, so they don't just diffuse on their own into the liver or so forth. So I believe that this holding tank concept is so good. And the peacekeeper cells that you can put with the islets is actually really important. Camillo Ricordi and others at the DRI work on MSCs, which is one of these cell types that seem to be hanging around and creating a peaceful environment. So these are good thoughts and a very good question. Yes, they need to be in context, most definitely. That's how ideally it has to be done.
Two more questions, and I thank you for that. Trying to address this without getting political, but a number of people have submitted the same question. If you could just speak at a high level to any concerns around the White House reductions in federal funding and how it may impact diabetes research in general.
So that, go for it, Dirk.
Go ahead, Dirk.
And of course, I think we all are very concerned. The NIH, the largest supporter of biomedical research in the world, lost about a quarter of their staff already. There are severely diminished opportunities for young scientists, for students, for postdocs, early career stage individuals, because they do not see a future in this country. So what will happen is the talent will walk away. They will find a place elsewhere. And we are really risking to severely damage the position that the US has attained as a premier place to conduct biomedical research at large. Science, as I try to outline a little bit the way I see it, that it progresses and is most effective. For it to work, it is vitally important to always recruit new talent. We have to incentivize young, smart, hardworking people to actually devote themselves to the field. There are no immediate, fame or riches in the field. This is driven mostly by passion, and it's hard work. But there are many smart young people who are willing to make sometimes these sacrifices if they can receive some assurances that their career paths are at least possible. If we don't get the best talent to join us in this effort, in particular, what we're talking about here, working towards better treatments, better prevention and cure reversal, perhaps of a clinical type 1 diabetes. We need to provide incentives, and they were traditionally provided here, and this is at risk.
I can get at this in a little bit more circumspect way, Michael. I think in general, having seen in our case, particularly the German funding system, because that's where I came from 35 years ago. I think we need to keep in mind that we need sustainability in our research systems. And NIH is only a part of that because they give out competitive grants. But competitive grants goes in peaks and valleys, and sustainability is extremely important for scientific careers and for core technologies. I think because of that, the DRI is such a unique place because we have the generous work of our foundation, and we are sort of in a way able to cover the valleys. And I think in other research systems like Germany, more of these valleys that are not covered, their core technologies that we need to maintain and can't lose, right? And careers who go over many years. To cover these valleys, other systems like in Germany have state and federal funds that are just given there. I think I feel that these vulnerabilities in the research system and the changes that are coming right now, they will reveal more of these vulnerabilities. And like Dirk says, we need to move forward in a way that we have a sustainable system. We also need a system where we have enough bandwidth to conduct solid science that is measured on gold standards. I strongly believe in that. Increase robustness of science and have incentives to increase cooperations just in addition to have individual investigator grants. So to make the long story short, I feel, of course, there's reason for concern, but in my mind, as always, any change also brings opportunities, and we keep the eyes on the ball, and need to see that we get eventually a cure for type 1 diabetes.
That's a perfect segue to our final question. Over the years, we have heard members of the scientific community reference anticipated timing for a cure for type 1 diabetes. When these timelines pass, they tend to diminish trust. What can you share from a scientific perspective, each of you, to rekindle our faith that a cure will ultimately be found?
So I think it's not binary. People have always painted a binary picture that a long time there's nothing happens and then all of a sudden something happens. While of course research progress is not linear, and Dirk spoke to that very well, you see that every single progress and very elegantly depicted by Dirk for the mRNA vaccines, every single progress is important, and it needs to also be celebrated, as we always say. And if you add them all up, a lot of little progress all of a sudden becomes a really big progress, rather than having the image saying, well, nothing happens, and then we all hope something big happens in the future. So I think we're shooting ourselves in the foot with this. We are actually making a lot of progress, even if it's small building blocks, like recently replacing in the immunosuppression regimen, and probably some of you know this, tacrolimus with anti-CD40 ligand. These are important building stones towards a progress for having more tolerable therapies and ultimately the replacement of beta cells. And we need to understand that that's how that evolves, rather than in a binary fashion. And with that comes that the prediction, when precisely you are approaching a state where you would say essentially the disease is cured, it's not a huge problem anymore, with all the progress that has been made. It's a process. It's something that evolves, rather than you have nothing and then you have a cure. I think that's a important narrative. That's how many things, minus very few exceptions, in medical and biological science have evolved. And I believe that that's an important story to tell for all of us researchers. We don't want to belittle our little progresses and say, oh, well, you just wait and then something big is going to happen. Definitely what comes though with it, the promising of timelines is really, it's not inherent of that process. That's not how that goes. You cannot say, oh, there's nothing happening, but I tell you for sure in 10 years it's going to be happening. There's things happening all the time. That's need to understand. And it's an exciting time where we are making a lot of progress in different areas, protecting the beta cells, replicating beta cells, having devices where you can have local immune suppression in the device, improving immune therapies. It's actually very, very exciting and cool times. So that's my opinion. Dirk, you go for it.
I'll keep it short. I agree with everything Matthias said. There's always room for improvement, and one area is how we communicate with the interested public. I think many of us are good at showing our passion, our excitement, but it's also important to convey a dose of humility and making clear what we do not know. And by being open and transparent and communicating to the public, to interested party, in a fashion that is not belittling or that promises something that we cannot possibly promise, but rather to invite an open, transparent discourse, trusting that sometimes explaining complex subject matters may take time, but the people in the audience here, you are all joining tonight because you have a genuine interest. And so I think we can, as scientists, we can do better in communicating both the excitement as well as the disappointments, the challenges, the little breakthroughs, what Matthias referred to as, we have to celebrate the little victories. Absolutely, we can communicate that to the public without overshooting the zeal, without implying that this is the solution to the future of humanity. So it is possible to improve the discourse that we are engaging in.
I thank you both. I'm going to turn it back over to Michael Burton now. Thank you, gentlemen.
Thank you, Michael, and thank you, Dirk. Thank you, Matthias. So I'll just close with a couple of observations really quick. And the first is a bit of inside baseball. And what I'll share with you is that we tend to keep a very close eye on, during a discussion like this, very close eye on the number of attendees at the start of this session and the number of attendees at the end of the session. There's virtually no difference in this discussion between the start and the finish. That is very unusual. And I think it points to the fact that it was a great discussion, and I'm sure the audience enjoyed it. I know I did. So thank you, Dirk. Thank you, Matthias, for your time and your expertise and your commitment to defeating this disease once and for all. And I also want to thank, too, those of you who have attended, who took in this discussion. I've noted a number of members of our national board, a number of members of our regional boards. Very grateful to them and to everybody who was on this call. You joined this call, as Dirk suggested, because you have been touched personally by this disease, every one of you in a different way. I can't tell you how grateful we are for everything you do to advance our mission. Simply put, we cannot do it without you. And especially in these uncertain times with regard to funding, your support, whether it's philanthropic or moral, is incredibly important, and we're grateful for it. So thank you very much for joining us. This is one in a part of a number of sessions that we do called Meet a Scientist. We will be having another session very soon. We hope you'll join us for that. And we look forward to keeping in close touch with you as we work to defeat this disease once and for all. So thank you and good night.
Bye.